Hello friends and welcome. In today's deliberation, we shall talk about disorders of lipoprotein metabolism, hypercholesterolemia, atherosclerosis, Alzheimer's disease. We'll talk about plasmalogen biosynthesis and its function, other ether and lipid-like platelet activating factors and its functions. The main objectives of today's deliberation are to understand the disorders of lipid metabolism, hypercholesterolemia, atherosclerosis, and Alzheimer's disease. To have general understanding of plasmalogen biosynthesis and its function, and finally to describe other ether and lipid-like platelet activating factors and its functions. Now let's have the general overview of the disorders of lipids. Fat absorbed from the diet and lipids synthesized by the liver and adipose tissue must be transported between the various tissues and organs for utilization and storage. Since lipids are insoluble in water, the problem of how to transport them in the aqueous blood plasma is solved by associating non-polar lipids, that is triacylglycerols and cholesterol esters, with amphipathic lipids and proteins to make water-miscible lipoproteins. In meat-eating omnivores such as humans, excess calories are ingested in the anabolic phase of the feeding cycle, followed by a period of negative caloric balance when the organism draws upon its carbohydrate and fat stores. Lipoproteins mediate this cycle by transporting lipids from the intestines as chylomicrons and from the liver as very low-density lipoproteins to most tissues for oxidation and to adipose tissue for storage. Lipid is mobilized from adipose tissue as free fatty acids bound to serum albumin. Abnormalities of lipoprotein metabolism cause various hypo or hyperlipoproteinemias. The most common of these is in the diabetes mellitus, where insulin deficiency causes excessive mobilization of free fatty acids and underutilization of chylomicrons and VLDL, leading to hypertriacylglycerolemia. Most other pathologic conditions affecting lipids transport are due primarily to inherited defects, some of which cause hypercholesterolemia and premature atherosclerosis. Obesity, particularly abdominal obesity, is a risk factor for increased mortality, hypertension, type 2 diabetes mellitus, hyperlipidemia, hyperglycemia, and various endocrine dysfunctions. Now let's talk about few diseases associated with lipids in greater detail. Let's first talk about hypercholesterolemia. It has been reported that absence of the LDL receptor leads to hypercholesterolemia and atherosclerosis. The results of Brown and Goldstein's pioneering studies of familial hypercholesterolemia reveal the physiologic importance of the LDL receptor. The total concentration of cholesterol and LDL in the plasma is markedly elevated in this genetic disorder, which results from a mutation at a single autosomal locus. The cholesterol level in the plasma of homozygotes is typically 680 mg per deciliter compared with 300 mg per deciliter in heterozygotes. A value of less than 200 mg per deciliter is regarded as desirable, but many people have higher levels. In familial hypercholesterolemia, cholesterol is deposited in various tissues because of the high concentration of LDL cholesterol in the plasma. Nodules of cholesterol called xanthomas are prominent in skin and tendons. Of particular concern is the oxidation of the excess blood LDL to form oxidized LDL. The oxidized LDL is taken up by immune system cells called macrophages, which become engorged to form foam cells. These foam cells become trapped in the walls of the blood vessels and contribute to the formation of atherosclerotic plaques that cause arterial narrowing and lead to heart attacks. In fact, most homozygotes die of coronary artery disease in childhood. A serum esterase that degrades oxidized lipids is found in association with HDL. Possibly, the HDL-associated proteins destroy the oxidized LDL, accounting for HDL's ability to protect against coronary disease. The molecular defect in most cases of familial hypercholesterolemia is an absence or deficiency of functional receptors for LDL. Receptor mutations that disrupt each of the stages in the endocytotic pathway have been identified. Homozygotes have almost no functional receptors for LDL, whereas heterozygotes have about half the normal number. Consequently, the entry of LDL into liver and other cells is impaired, leading to an increased plasma level of LDL. Furthermore, less ideal enters liver cells because ideal entry too is mediated by the LDL receptor. Consequently, IDL stays in the blood longer in familial hypercholesterolemia, and more of it is converted into LDL than in normal people. All deleterious consequences of an absence or deficiency of the LDL receptor can be attributed to the ensuing elevated levels of LDL cholesterol in the blood. Now let's talk about the atherosclerosis in detail. Atherosclerosis, hardening and narrowing of the arteries, silently and slowly blocks arteries, putting blood flow at risk. It's the usual cause of heart attacks, strokes and peripheral vascular disease. These are together called as cardiovascular disease. The disease disrupts the flow of blood around the body, posing serious cardiovascular complications. Arteries contain what is called as endothelium, a thin layer of cells that keeps the artery smooth and allows blood to flow easily. Atherosclerosis starts when the endothelium becomes damaged, allowing LDL cholesterol to accumulate in the artery wall. The body sends macrophage white blood cells to clean up the cholesterol, but sometimes the cells get stuck there at the affected site. Over time, this results in plaque being built up consisting of bad cholesterol, that is LDL cholesterol, and macrophage white blood cells. The plaque clogs up the artery, disrupting the flow of blood around the body. This potentially causes blood clots that can result in life-threatening conditions such as heart attack, stroke, and other cardiovascular diseases. Causes of atherosclerosis. Atherosclerosis is caused by macrophage white blood cells and fat that accumulates in arteries. The white blood cells are originally sent by the body's immune system to clean up LDL cholesterol pockets. When they stick to an artery, they secrete a molecule called natrin-1. This stops normal migration of the macrophages out of the arteries. As a result, what you have left is a mixture of clumped up cholesterol pockets and white blood cells. This is the plaque that can disrupt blood flow. Certain factors that can damage the inner area of the artery, that's endothelium, and can trigger atherosclerosis include high blood pressure, high levels of cholesterol, smoking, high levels of sugar in the blood. Areas of the artery that are damaged are likely to have plaque build up which can eventually break open. When the plaque breaks open, blood cell fragments called thrombocytes accumulate at the affected area. These fragments can then stick together, forming blood clots. Diabetes. Patients with poorly controlled diabetes who frequently have excess blood glucose levels are much more likely to develop atherosclerosis. Genetics. People with a parent or sibling who has atherosclerosis and cardiovascular disease have a much higher risk of developing atherosclerosis than others. Another factor is air pollution. In 2007, researchers from the University of California in Los Angeles linked exposure to diesel exhaust particles in air pollution to a higher risk of bad cholesterol buildup in the arteries. It's now time to talk about another disease which is related with lipids, that's Alzheimer's disease. Alzheimer's disease is the most common neurodegenerative disorder affecting over 36 million people worldwide. Alzheimer's patients suffer from dementia and memory loss and go through a progressive, irreversible, and eventually fatal loss of cognitive functions. When Alzheimer firstly described the disease over 100 years ago, he identified abnormal protein deposits as well as fat tissue inclusions and altered lipid composition in the brains of his patients. These observations suggested a possible relation between Alzheimer's disease and lipid imbalance, which was established decades later when the strongest genetic risk factor in Alzheimer's disease was linked to apolipoprotein E, the major lipid transporter in the central nervous system. Presently, it's well known that cholesterol and many other lipids are involved in various ways in key aspects of the disease. For example, lipids strongly interfere with a protein crucial to Alzheimer's disease called amyloid beta precursor protein. APP is located inside the fatty membranes surrounding nerve cells and can be cut into smaller protein segments called amyloid beta. This smaller protein is very unstable and easily aggregates into clumps that can become large and sticky. Depositing as plaques in the brain, some of those proteins clumps can also remain as small, free and permeable structures easily interacting with fatty membranes and interfering with synapses and cell-to-cell communication in the brain. The potentially toxic process of amyloid beta formation and aggregation occurs preferentially in lipid rafts, which are regions of fatty membranes that are rich in cholesterol and a particular type of lipid named gangliosides. Cholesterol plays a major role here as it binds APP, promoting its localization in lipid rafts and helping other molecules to cut APP into the toxic amyloid beta. The lipid rafts are also believed to serve as templates for amyloid beta aggregation because lipid-rich environments can change the structure of amyloid beta, turning it into a more aggregation-prone molecule. Moreover, the toxic action of small amyloid beta aggregates is thought to include the formation of pores in the membranes of brain cells. These holes perturb the flow of ions and other vital molecules in and out of the cells and can lead to cell death. Cholesterol may importantly influence the insertion of amyloid beta into the cell membranes, its aggregation and pore formation. Although the molecular role of cholesterol in Alzheimer's disease is not entirely clear, clinical evidences indicate that cholesterol has a major influence on the disease progression. In fact, elevated blood levels of cholesterol are a risk factor for Alzheimer's disease, and cholesterol-rich diet promotes accumulation of amyloid beta inside the cells and increases its cerebral load. Cholesterol levels in the human brain were also reported to increase in the early stages of the disease. This apparent connection between the elevated blood cholesterol and the Alzheimer's disease raised the possibility that cholesterol-lowering drugs may benefit the patients. Furthermore, some cell and animal model studies report that cholesterol-lowering drugs can reduce levels of amyloid beta in brain cells. Limited clinical studies report decreased risk of Alzheimer's disease or improved cognitive function in patients. However, more studies are needed to draw unequivocal conclusions about possible role of lipid-lowering drugs in Alzheimer's treatment. Moving on, now let's talk about the biosynthesis of glycerol, ether phospholipids. This pathway is located in peroxisomes. Dihydroxyacetone phosphate is the precursor of the glycerol moiety of glycerol, ether phospholipids. This compound combines with acyl-CoA to give 1-acyl dihydroxyacetone phosphate. The ether link is formed in the next reaction, producing 1-alkyl dihydroxyacetone phosphate, which is then converted to 1-alkyl glycerol 3-phosphate. After further acylation in the 2 position, the resulting 1-alkyl 2-acyl glycerol 3-phosphate is hydrolyzed to give the free glycerol derivative. Plasmalogens, which comprise much of the phospholipid in mitochondria, are formed by desaturation of the analogous 3-phosphoethanolamine derivative. Platelet activating factor is synthesized from the corresponding 3-phosphocholine derivative. It's formed by many blood cells and other tissues and aggregates platelets at concentrations as low as 10 raised to the power minus 11 moles per liter. It also has hypotensive and ulcerogenic properties and is involved in a variety of biologic responses including inflammation, chemotaxis, and protein phosphorylation. Moving on now, let's have the general understanding of plasmalogens. Plasmalogens constitute as much as 10% of the phospholipids of brain and muscle. Structurally, the plasmalogens resemble phosphatidylethanolamine but possess an ether link on the SN1 carbon instead of the ester link found in acylglycerol. Typically, the alkyl radical is an unsaturated alcohol. In some instances, choline, serine, and inositol may be substituted for ethanolamine. Significant branch points in the pathway occur at the phosphatidate and diacylglycerol steps. From dihydroxyacetone phosphate are derived phosphoglycerols containing an ether link, the best known of which are plasmalogens and platelet activating factor. Glycerol 3-phosphate and dihydroxyacetone phosphate are intermediates in glycolysis, making a very important connection between carbohydrate and lipid metabolism. Now let's talk about the Glycerol phospholipids having ether-linked fatty acids. Some animal tissues and some unicellular organisms are rich in ether lipids, in which one of the two acyl chains is attached to glycerol in ether rather than ester linkage. The ether-linked chain may be saturated as in the alkyl ether lipid or may contain a double bond between C1 and C2 as in plasmalogen. Vertebrate heart tissue is uniquely enriched in ether lipids. About half of the heart phospholipids are plasmalogens. The membranes of halophilic bacteria, ciliated protists and certain invertebrates also contain high proportion of ether lipids. The functional significance of ether lipids in these membranes is unknown. Perhaps their resistance to the phospholipase that cleave ester-linked fatty acids from membrane lipids is important in some roles. At least one ether lipid, platelet activating factor, is a potent molecular signal. It's released from leukocytes called basophils and stimulates platelet aggregation and the release of serotonin, a vasoconstrictor. Now at last, let's talk about the plasmalogen synthesis, which requires formation of an ether-linked fatty alcohol. The biosynthetic pathway to ether lipids, including plasmalogens and the platelet activating factor, involves the displacement of an esterified fatty acyl group by a long chain alcohol to form the ether linkage. Head group attachment follows by mechanisms especially like those used in formation of the common ester-linked phospholipids. Finally, the characteristic double bond of plasmalogens is introduced by the action of a mixed function oxidase similar to that responsible for desaturation of fatty acids. The peroxisomes is the primary site of plasmalogen synthesis. So students, we in this lecture talked about the various lipid disorders and biosynthesis of plasmalogens and platelet activating factor. With that, we come to the end of today's program. Hope you have understood it well. Thank you and goodbye.